amazing video. Thank you guys so much for that. My name is Erwin Tanner. I am a nurse practitioner at Kennedy Krieger Institute, and I will be moderating this next portion. Thank you so much for having me this morning. It is with great pleasure that I introduce Dr. Horvath. She is a clinician, academic, and trained neurologist. Her work has advanced the understanding, diagnosis, and treatment of mitochondrial translation deficiencies and led her to her current position of Director of Research at the Department of Clinical Neurosciences at the University of Cambridge. We are looking very forward to your presentation. Thank you so much, Dr. Horvath.
Thank you very much for the invitation to present at this conference. And it was really amazing to see these videos and see how well your children are doing. And I am very happy to participate in this work. And I will try to share my presentation. Do you see it now? Okay. So I am a neurologist, and I have been working on the diagnosis, on the research, and even the management of patients with mitochondrial diseases over 20-25 years. When I was working still in Munich, Germany, in the diagnostic lab, I realized that there were patients, many patients, usually children, affected mitochondrial tRNA synthetases. So it seems that mitochondrial tRNA synthetase defects are relatively frequent in causes of combined respiratory chain deficiency. There are over 50 genes known so far, which are affecting the transcription or translation of the mitochondrial DNA. And many of these genes are causing severe multisystem infantile diseases, and some of them are extremely rare. However, the diseases caused by mitochondrial tRNA synthetase defects are relatively frequent, and they have very tissue-specific clinical presentation. A few years ago, we did a review on the role of tRNA synthetases in neurological disease, and you can see at this picture, and there was a very nice introduction to the tRNA synthetase field for Dr. Fatami before. So then you understand already that there are tRNA synthetases which are responsible for the mitochondrial tRNAs, and other tRNA synthetases are responsible for the cytosolic translation. And what we showed that most of these genes have tissue-specific presentation, and the main tissues which are affected are the nervous system, heart, skeletal muscle, or liver. There are some, even within the neurological presentation, there are some presentations which are more common than others, and there are some of the genes which have very characteristic cell type specific neurological presentation. For example, some of the cytosolic tRNA synthetases and the so-called bifunctional tRNA synthetases, which are encoding both the cytosolic and mitochondrial tRNAs, dominant mutations in these genes can cause Charcot-Marie-Tooth peripheral neuropathy, while the mitochondrial tRNA synthetases are recessive, or most of the other tRNA synthetases are also recessive, and some of the mitochondrial tRNA synthetases have very specific presentations in the white matter, so some certain cell types are affected and they cause leukoencephalopathy, or some others cause epileptic encephalopathy or hearing loss or intellectual disability. So all these enzymes, these tRNA synthetase enzymes, are present ubiquitously in each cell, and it is not understandable why they would cause so tissue-specific presentations. The role of the tRNA synthetases are to add the specific amino acid to the tRNAs, and it is also no reason why would it be any different in the different cell types, but as you can see, some of the mitochondrial tRNA synthetases are affecting the brain, others the heart or kidney or the skeletal muscle. So I will present you some of the most common clinical presentations of the tRNA synthetase mutations. One of them is Perrault Syndrome, which is a combination of sensorineural hearing loss and premature ovarian failure. So as you can see in this large pedigree, that the females have both hearing loss and ovarian failure, and the male individuals have hearing loss. And interestingly, many of these cases can be caused by mutations in LARS2 or HARS2, but there are also other nuclear genes which are affecting mitochondrial translation, which can also cause Perrault syndrome. However, some of the mutations in LARS2 have been shown to cause a much more severe infantile multisystem disease. Another interesting phenotype is mitochondrial myopathy, lactic acidosis, and sideroblastic anemia, so-called MLASA, which is caused by mutations in the YARS2 or PUS1 gene. And in addition to these symptoms in the YARS2 mutations, the patients may have hypertrophic cardiomyopathy, so heart problems, and respiratory failure. But these patients do not have any brain involvement at all. And there are some founder mutations in the Middle Eastern and Scottish population. So in those populations, YARS2 defect is more frequent. Another mitochondrial tRNA synthesis defect, RARS2, is particularly affecting the cerebellum. You can see on the pictures how much smaller is the cerebellum in some patients than compared to controls. And in this condition, there is usually the respiratory chain defect affects several organs. While we have already heard a bit about DARS2 mutations in the first presentation, so these cause a very characteristic MRI presentation, and even the MRI syndrome has been reported before the gene was found, and it was helping the identification of the gene. And these patients have a leukoencephalopathy with brainstem and spinal cord involvement and a high lactate. Some patients with this condition have a severe early onset, others have a milder adult form. But the most common presentation is a childhood onset, slowly progressive paresis and ataxia. And there are many pathogenic mutations reported in the DARS2 gene, but the majority of the mutations actually affect splice sites, which is an interesting avenue to develop treatments for some of these mutations with different approaches targeting splicing, such as antisense oligos or other ways. And probably we will hear about it in some later presentations. ARS2 mutations can have two different clinical presentations. So there is one common, relatively common mutation in the position 592 in ARS in the editing domain, which is mostly associated with the heart involvement. So these patients have cardiomyopathy, while mutations in the other domains of the ARS protein and gene more likely to cause leukoencephalopathy with ovarian failure. And the ovarian failure is a little bit linking it with Perrault syndrome. And it's not quite clear at the moment what is the reason for this tissue specificity of the different mutations in this gene, but it is possible that it is linked to the localization or the severity of the mutation. Another very characteristic MRI finding was reported in mutations in the ERS2 gene, which is mostly affecting the white matter. So it was in leukoencephalopathy with thalamus and brainstem involvement and high lactate. However, there were some patients. So we had a patient who had a multi-system, very severe infantile disease and did not develop this typical MRI signs, but died very early. And it seems that about 1/3 of the patients with ERS2 mutations have a severe infantile progressive course, but about 2/3 of the patients have a kind of partially reversible disease course, and they can show improvement. And this is something what is very interesting, and it may link it with some other reversible mitochondrial diseases, which I will mention later. And also there are other mitochondrial tRNA synthetases which do not have a characteristic phenotype or MRI presentation like TARS2. In this recent paper, we had one patient in this cohort. The 5 patients had very different clinical presentations, including mitochondrial encephalomyopathy, dystonia, epilepsy, hearing loss, hyperhidrosis, or learning difficulties, but the MRI did not show characteristic findings. So I showed you that there are some neuronal cell types are more affected in some of the tRNA synthetase mutations. Therefore, we decided to study some neuronal cellular models of mitochondrial tRNA synthetase mutations. Juliane Müller and Oliver Podmanitsky are working on this project. Oliver is a PhD student whose project is particularly on mitochondrial tRNA synthetases. So first we used a relatively simple method to convert fibroblasts of patients directly into induced neuronal progenitor cells without making iPSCs in between. And these induced neuronal progenitor cells can be further differentiated into neurons. And we made the differentiation into 4 brain type neurons, but it would have been possible to go into motor neurons, oligodendroglial or astrocytes. But these are what we noticed in our methodology. They are more difficult to manage from these induced neuronal progenitor cells. And in the meantime, we switched our research more into iPSC-based neuronal differentiation. But I will now show you the data what we obtained on the induced neuronal progenitor cells. Oh, sorry, it went back. Okay. So mitochondrial diseases are often tissue specific, and the fibroblasts often do not express the mitochondrial defect. And it was the case also with some of our mitochondrial tRNA synthetase cell lines. However, the induced neuronal progenitor cells are more close to neurons, and they showed more of a defect than the fibroblasts. And when we differentiated them into neurons, the neurons show more significant defect of the mitochondrial complexes. So they are more sensitive of the defect. So it's a good model to study the effect of these mutations. We performed transcriptomic studies and proteomics on these IMPC cellular models, and we are currently analyzing these data to identify important pathways which are important in the pathomechanism of these diseases. And there are some pathways that we have already identified. For example, the role of the integrated stress response, which was activated in some of the IMPC cell lines, but it has also a very important role in skeletal muscle. So we are pursuing, Denisa Hathazi, a postdoc in my lab, is pursuing the role of the integrated stress response in some mitochondrial translation defects. And why it is an important avenue, because there are some small molecules which you can use to target the integrated stress response. So it would provide some potential treatment for some of the mutations. We also studied the relationship between mitochondrial translation and cytosolic translation. And it is showing our controls and the ARS2 IMPC cell line. And what we found that the rate of the cytosolic translation in this mutant cell is not too much different from the controls. However, the pattern of the translation, a pattern of these bands on the puromycin incorporation assay showed some differences, and we are currently looking into these differences as well. And as I mentioned you, so I cannot show too much data because we are working on this project, and we moved over to iPSC-derived neuronal cell lines and also to an organoid model, and hopefully we can present more data on these models later on. In addition to the in vitro models, we also use zebrafish as an in vivo model of mitochondrial diseases. And Ben Munro in my laboratory generated an ERS2 knockout or knockdown zebrafish model with the CRISPR-Cas9 mutagenesis. So this zebrafish has a homozygous frameshift mutation in the aaRS2, but it's viable. So it has a shorter lifespan. It lives just a few weeks long, but it is viable, which is a very important point because it is very difficult to generate mouse models of knockout of the different mitochondrial tRNA synthetase defects. So it seems that the zebrafish is a good animal model to study this condition. And Oliver has been studying this zebrafish already, and we'll further continue to characterize this zebrafish and do further studies. But we already show that this zebrafish has a decrease in the mitochondrial respiratory chain complexes 1 and 4, an increase in complex 2. So it is a very good representation of the mitochondrial translation defect. And this fish can be further studied to study the effect of this variant in the different organs and tissues in the fish. And also we can use it for supplementation studies with small molecules. So far I was talking about the autosomal recessive effect of the mitochondrial tRNA synthetase mutations, but there is also another aspect of the mitochondrial tRNA synthetases which we were working on quite intensively over the past few years, and I would like to present some of these data also. So in some families who carry mitochondrial tRNA mutations, we noticed that if there is an additional variant, a heterozygous variant in some of the mitochondrial tRNA synthetase gene, the two variants, the mitochondrial tRNA variant together with the tRNA synthetase variant can have a pathological effect. And this is a kind of digenic inheritance of the mitochondrial DNA and the mitochondrial tRNA mutation. And a very good example for it is a very rare reversible mitochondrial disease, the reversible infantile COX deficiency myopathy. So in this family, this boy was very weak and very severely affected in the first days of life. He needed mechanical ventilation, tube feeding, and he spent the first 6 months of his life at the intensive care unit. However, then he spontaneously started to get better, and then he had a delayed motor development, but he has a normal life now. The muscle biopsy in the 3 months of age showed very severe COX deficiency and mitochondrial abnormalities, but it also improved in parallel with the clinical recovery. And his brother and his maternal aunt and her twin brother also had the same reversible symptoms in infantile period. And in this family, we identified a homoplasmic mitochondrial tRNA glutamic acid mutation in this position, which was the cause of the disease in this family and in also many other families worldwide. So that's homoplasmic mutation is causing this disease. However, as you can see in this family tree already, that not all homoplasmic mutation carriers develop the symptoms. So I visited this family a few years ago again, and as you can see, there are these 3 family members who had the symptoms, and although all the other family members who you see in the picture carry the homoplasmic mitochondrial DNA mutation, only these 3 people develop the symptoms. So to answer the reason why it is the case and what else is needed to develop symptoms in this mutation, we performed whole exome sequencing in 19 families who were all homoplasmic for this mutation. And interestingly, in 6 out of these families, we detected heterozygous variants in the aaRS2 gene in the affected individuals. Some of these variants were already reported in recessive families with the leukoencephalopathy phenotype, and so they were definitely pathogenic. However, the heterozygous parents who were the carriers of these variants do not show symptoms. So it seems that to have symptoms, you need to carry both the homoplasmic mitochondrial DNA variant and the heterozygous variants in the aaRS2 gene. And it explained the rarity of the reversible COX deficiency myopathy because the frequency of the homoplasmic mitochondrial DNA mutation would be 1 in 10,000 in the European population. So it would need to be many more patients with this disease if all patients develop symptoms. However, if the carriers who also carry a heterozygous variant in ERS2 or in other families in other genes. These are the ones who develop the symptoms only. This is explaining the rarity of this disease and explaining that it is actually a digenic disease caused by both the mitochondrial and the nuclear DNA mutation. Just again to show the fact that this mutation, which is causing the reversible Cox defect, is exactly at the very end of the tRNA molecule, where the amino acid is being added by the ERS2 enzyme. So a mild defect of the tRNA and a mild defect of the ERS2 together can cause a more severe defect. This picture is illustrating our cohort of 19 families. So in the 19 families, in 6 families, we detected heterozygous variants in the ERS2, which would trigger the phenotype, and in some other families in other similar genes affecting the tRNA function, which is very important in the genetic counseling of these families and patients. There are other examples of the interaction of the mitochondrial tRNA variants with the mitochondrial tRNA synthetase variants, because in this paper it was reported for a mitochondrial tRNA valin mutation, the penetrance was influenced by a heterozygous VARS2 mutation. But I find it very interesting to further observe the interaction of the tRNA mutations with the tRNA synthetase mutations. And the clinical geneticist, fellow Katarina Olimpio in my lab, is looking into this aspect, and we are trying to understand it further in larger patient cohorts. There was another interesting discovery, what we found in this reversible Cox disease, and it was the fact that we detected a very important role of the amino acids in these patients. So in the beginning of the disease, if an infant carries both the mitochondrial and the tRNA synthetase variant, there was a very severe translation defect. And this defect activated the integrated stress response. And also it did a metabolic shift and activated a lot of amino acid transporters and the activation of the fatty acid oxidation. So it led to increased level of amino acids in the cell. And also the original translation book activated the mTOR pathway, which then led to increased mitochondrial biogenesis, and the mtDNA content and the level of the tRNA was also increased, and that resulted in the recovery and the resolution of the translation defect. But what was very important, that the integrated stress response and the amino acids, and particularly the cysteine, has a very important role in infants, are something what we may use to treat not only this condition, but maybe other mitochondrial conditions. And regarding the role of cysteine, there was other indication in other reversible infantile mitochondrial disease, in reversible infantile hepatopathy, that the reason why these patients develop the symptoms is infantile periodis, that the dietary intake of cysteine has to be relatively high in infants, because the cysteine is an essential amino acid. It cannot be made from other amino acids in the infantile period. So it has been already proven that cysteine supplementation is very beneficial for some patients with the reversible infantile hepatopathy, because it can prevent the liver transplantation, or it can improve the mitochondrial translation in these patients. And we also showed in our cellular models that if we deplete the cysteine in the cell culture medium, some cells develop mitochondrial translation defect. And if we supplement the cysteine again, we can improve the mitochondrial translation defect in some selected cell types, and then some selected mitochondrial translation defects. And so based on this data, we decided to study whether supplementation of cysteine or other amino acids, or modifying the integrated stress response with some small molecules, can be beneficial in other mitochondrial translation defects. And in fact, we did some studies in fibroblasts of the very common 3243 and A344 mutations, and we showed improvement in the mitochondrial function after cysteine or N-acetylcysteine supplementation. So we are currently further investigating whether cysteine or other amino acids could be beneficial for other mitochondrial ARS defects. And there was a very interesting paper published just very recently from a Dutch group, which showed that supplementation with the cognate amino acids in 4 different cytosolic tRNA synthetases actually improved the symptoms in the patients. And in these cases, the defect was had a loss of function effect of the enzyme. And actually, most of the mitochondrial tRNA synthetase defects have a loss of function effect. So it may be possible that with amino acid supplementation of the cognate amino acid or maybe other amino acids can improve mitochondrial tRNA synthetase mutations also. And we are currently also trying to look into this in our cellular and zebrafish model. So at the end, just for a summary, how can we explain the tissue specificity of the mitochondrial tRNA synthetase mutations? So one question was originally that is the clinical presentation of the tRNA synthetase similar to the mutations in the cognate tRNAs, but there is no indication in the literature on this. Or it was a question also whether the expression of the tRNA synthetase would be different in tissues, but it is also not the case. The amount of amino acids which are encoded, which are transported with the help of the tRNA synthetases is also not particularly different in the different tRNA synthetases. And it is possible that the availability of amino acids can be different in some organs. And we are looking into the amino acid supplementation rescue in mitochondrial tRNA synthetases. And it is very important to observe the diet or the information which is available already in patients who may take amino acid supplementation. So we would be very much interested to look into this data, which are available already in the patients to prove or disprove this theory. And also it's possible that there are non-canonical function of some mitochondrial tRNA synthetases. It is very well known for cytosolic tRNA synthetases that non-canonical functions play a role in inflammation, infections, infection, and even cancerogenesis. So it is possible that there are other functions of mitochondrial tRNA synthetases which we don't know. And also we are investigating the link between the mitochondrial and cytosolic translation. And also it is possible that the non-cognate, so some tRNA synthetases can compensate the defect of other tRNA synthetases in some tissues. And there are all questions which would be good to answer with the research. And I would just like to thank the people in my lab and who contributed to the research in mitochondrial tRNA synthetases and also to all my collaborations all over the world. And I thank you, and also the funders, and thank you very much for your attention.
Thank you so much for your time, Dr. Horvath. That was great. After the next 2 presentations, there will be an opportunity for questions. So just a reminder, please write your questions within the Q&A function, and we will get to them after 2 more presentations. So Nadine, do you mind if you could please pause the recording for our next speaker? I'm a professor in genetics at the Department of Chemistry, Life Sciences, and Environmental Sustainability at the University of Parma. Her research activities focus on the use of yeast as a model system to study mitochondrial disorders. Thank you so much.
Good morning to everyone. First of all, I would like to thank the organizers for giving me the great opportunity to present my work and to show you the potentiality of the yeast Saccharomyces cerevisiae as a model system. So now I will share with you my presentation. Okay, so let's start. Probably most of you are not familiar with Saccharomyces cerevisiae, commonly known as baker's yeast. It's a simple unicellular organism, but it has proven to be a very useful model organism, in particular to study mitochondrial function. Its simplicity is, in my opinion, Probably most of you are not familiar with Saccharomyces cerevisiae, commonly known as baker's yeast. It's a simple unicellular organism, but it has proven to be a very useful model organism, in particular to study mitochondrial function. Its simplicity is, in my opinion, a great advantage as it allows to obtain results in a short time and low cost. Moreover, genetic manipulation is easy and a lot of tools for genetic engineering are available. As it is an eukaryotic cell, yeast shares highly conserved molecular and cellular mechanisms with human cells, including mitochondrial biogenesis and function, and of course, tRNA aminosylation, that is the topic of this symposium. Of course, it also has some limits. For example, not conserved genes and pathways cannot be studied in yeast. And tissue-specific phenotypes found associated with mutation in different mt-ARS cannot be assessed. So what can yeast do for patients? First of all, it can contribute in diagnosis. Today, one of the challenges in the field of mitochondrial diseases is identification of the genetic basis of the conditions of patients lacking a diagnosis, generally because of the clinical and genetic heterogeneity of mitochondrial diseases. Thanks to the extensive use of next-generation sequencing, an increasing number of variants have been identified in mt-ARS gene. Since most of these variants are private or sporadic, it is crucial to assess their causative role in the disease by functional analysis in model systems. So, in other words, it is necessary to validate these new variants. Functional analysis could be performed in fibroblasts from patients, but they often did not show any easily measurable readout. On the contrary, mutations in genes encoding for mitochondrial functions result often in a clear and distinguishable respiratory phenotype in Saccharomyces cerevisiae. That, for this reason, has been extensively used to study the consequences of potential pathological human mutations. I don't have the time to get inside into the specific techniques used to create suitable models. Briefly, what we do is that the wild type and the mutant allele equivalent to the alleged pathological allele are introduced into a strain deleted of the corresponding ortholog gene, and then a complementation assay is performed. So I have studied several mutations in 7 different genes coding for mt-ARS. And today I will show you as an example the case of ARS2, which was my first mt-ARS gene studied. ARS2 encodes for the mitochondrial alanyl tRNA synthetase. The patient presented leukoencephalopathy, and next-generation sequencing revealed compound heterozygous mutations. So one mutation was a missense mutation, and the other one was a nonsense mutation in the last part of the portion of the protein. Because no biochemical readout was detected in patient cells, we tested the possible deleterious impact of the ARS2 mutation in Saccharomyces cerevisiae using the yeast ortholog gene ALA1. The construction of this specific model is a little bit complex, but in the end, we obtained yeast strains expressing alleles carrying the mutations equivalent to those found in patients. So the question is now, do these mutations lead to mitochondrial dysfunction? So in other words, is the yeast carrying these mutations sick? Validation or not of an alleged pathological mutation is based in yeast on a very quick and simple test, that is a simple grow test. So the wild type Saccharomyces cerevisiae is able to grow both on fermentable carbon sources, such as glucose, and on non-fermentable or oxidative carbon sources, such as glycerol. On the contrary, mutations affecting mitochondrial function lead to a partial or total oxidative growth impairment. So here you can see the results we have obtained. In green, you can see the wild type strain. And as expected, it is able to grow both on glucose and on glycerol. In red, you see the null mutant, so the mutant without the gene, and as expected, it's completely unable to grow on glycerol. And here in blue, you can see the mutants under investigation. So the nonsense mutation leads to a very severe phenotype similar to the null mutant. On the other hand, the missense mutation here seems not to be detrimental. As you can see, the growth is similar to the wild type's growth. But if we repeat this experiment in a more stringent condition, so at high temperature, we can observe a reduced oxidative growth. So this mutation is thermosensitive. Conclude that both mutations are detrimental in yeast, thus validating them as pathological in humans. To better quantify the defects, we have also measured oxygen consumption rate. As you can see, we have obtained similar results as oxidative growth. So one mutation is very severe, leading to a complete loss of function of the protein, and the other mutation is leaky, indicating that the protein function is partially maintained. So what can yeast tell us about a mutation? Yeast can tell us if a specific mutation is detrimental or not. An important advantage of using yeast is that it is possible to determine the consequences of a single variant in an identical genetic background, in contrast to patients who are genetically heterogeneous. So in the ARS2 example, we have seen that the two variants individually are detrimental. But in many cases, patients are compound heterozygous. So in yeast, we can evaluate the contribution of each mutation to the pathological phenotype. So in the ARS2 example, we have seen that one was a null allele and the other was a hypomorphic allele. Another interesting aspect is the severity of the disease. So given the clinical heterogeneity of mitochondrial disorders, predicting disease severity in yeast could be challenging, but we have observed empirically that often the severity of the impairment in yeast correlates with the severity of the clinical phenotype of the patient. So in the ARS2 example, we have seen that the F22C mutation was associated in yeast to a leaky thermosensitive phenotype in agreement with the slow disease progression in the patient. What else can yeast do for patients? Besides validation, yeast can also be used to determine the pathogenic mechanism behind a mutation. Mutations in proteins involved in aminoacylation of mitochondrial tRNA can result in defects of mitochondrial protein synthesis, MPS. So we assess if the OXPHOS defect that we have observed was due to a defective MPS. As you can see, mitochondrial protein synthesis is completely abolished in the strain expressing the nonsense mutation and is significantly decreased in the strain expressing the missense mutation only in stressing conditions. So to evaluate whether the defective mitochondrial protein synthesis is due to the reduced level of aminoacylated tRNA, we evaluated the charging of tRNA with alanine. So the analysis revealed that the nonsense substitution completely inhibit the charging of mt-tRNA, whereas the missense mutation determined a partial reduction in tRNA loading and only at high temperature. So overall, the results obtained in yeast point out that the molecular mechanism underlying leukoencephalopathy is the charging defect. This in turn leads to the reduction of protein synthesis and then to a reduction of respiratory complex activity, and then to a reduction of oxygen consumption rate, and finally to the oxidative growth defect that we initially observed. Another major challenge regarding mitochondrial diseases is the availability of pharmacological treatment that is still insufficient. So can this give a contribution to the identification of new treatment? And my answer is yes. So substrate enhancement, which in some cases can be obtained through dietary supplementation, is an emerging treatment for some mitochondrial pathologies. So it is possible that an increased availability of the amino acid substrate for a given mt-ARS could improve the catalytic efficiency of the mutant enzyme. So we have tested this hypothesis in some yeast models. Here you can see the results about some mutations in the tyrosine tRNA synthetase. As you can see, tyrosine supplementation here is indicated in red compared to the strains that are not supplemented with tyrosine that are indicated in blue. So tyrosine supplementation leads to an increase in the respiratory activity of leaky mutant. So when the enzyme is partially functional, tyrosine supplementation can be beneficial. On the contrary, when the enzyme activity is strongly affected, the supplementation is not effective. So a beneficial effect of amino acid supplementation has been observed also in other yeast models. However, the approach did not always produce the same beneficial effect, suggesting that the response to amino acid supplementation could be gene-specific or even mutation-specific, and it is hardly to predict in simple models. However, if a beneficial effect is observed in yeast, it is certainly worth to experiment amino acid supplementation in patients. And what about identification of new pharmacological treatment? Can this help in this? So in recent years, high-throughput phenotype-based screening have been developed in yeast to identify potential therapeutic drugs. In particular, a drug repurposing approach, that is the investigation of existing drugs for new diseases, can be used. And in particular, it is possible to use chemical libraries containing FDA-approved drugs, thus allowing a reduction in time and cost, since the safety of these drugs have been already assessed. Here I show you a very preliminary and unpublished data obtained on the ARS2 model. So how does this screening work? The mutant strain is spread on a solid medium in condition where it is unable to grow. And then it is exposed to compounds that are spotted on small filters. The appearance of a halo of enhanced growth around the filter indicates that the drug is beneficial for yeast. And then this drug is considered potentially therapeutic for patients. For now, I have tested about 100 drugs, and 3 were able to ameliorate the mutant's phenotype. Of course, further work will be necessary to confirm these results, including testing the efficacy of these drugs in other model systems. So we will see if these drugs will be good candidates or not. So I hope I have convinced you that yeast, despite its simplicity, can be helpful for mitochondrial medicine. Here you can find my email, so feel free to contact me if you have questions or if you would like to explore the possibility to study some new variants in yeast. Finally, I would like to thank all my colleagues and all my collaborators, and of course, you for your kind attention.
Thank you. David, I will turn this slide deck back over to you, please.
Amazing. Thank you so much for all of the presenters this morning. Appreciate your guys' time and effort. And I just wanted to confirm with Desiree, are you wanting to pause the recording for the Q&A?
Yes.
All right. Nadine, if you don't mind pausing the recording, please. And
my name is Terry Sanger, and I'm the Chief Scientific Officer, and I'm a child neurologist. This is a very exciting place to be. I think we are a hospital that really has this strong vision that we want to help children, and we'll do it any way we can. We're not mired in some tradition that says you have to do it this way, you have to do it that way. We're careful, we care deeply about our children, we will make sure that people are safe, we'll make sure things are done ethically, but I think there's tremendous opportunity to create things that have never been created before. I think the growth potential is huge. For the bench science, we need to build more space. Over the next 5 years, I'm expecting to have significantly more space available. We already have a plan to double our wet bench space. We are significantly expanding our ability to support clinical trials with additional clinical research coordinators. We are increasing our data science support so that we can rapidly search the electronic health record, being able to provide these sort of 21st century technologies and the people to support them is very important. And the relationships that I'm hoping to form with our university partners, we can provide a safe environment in which good research can be done by this very large community of very dedicated and talented people that's at UC Irvine. So by close collaborations with UC Irvine, we gain a lot. There's a lot of institutions that I've worked in where you sort of can't see the forest for the trees. You start to think that the procedures are the important thing or the rules are the important thing. And it's not, it's the children are the important thing, and I think everybody here sees that. I've never seen anything like this. I've never seen a hospital that has this kind of drive to be better and drive to be open and drive to ask questions and always focused on making children better.